we're going to see adaptation. And we can see a pretty profound adaptation. And if any of you have ever, been involved in the rehab process or the training process of athletes, not everybody sees this. This is a mean response. Some people see 15%, some see 2%, and part of you start talking about personalized responses in personalized medicine, there may be some of the signaling differences between people that are driving some of this. Okay, so, we start talking about long-term resistance exercise. We see increases in individual fiber cross-sectional area, and we tend to see larger increases in the fast twitch type versus the slow twitch type muscle fibers. So we're really talking about different adaptations when it comes to muscle fiber types, and then when you compare that to what we learned about endurance exercise, we're going to start seeing differences in the molecular pathways between these fiber types. Distribution between type 2 and type 1 really pretty much unchanged. However, we start to see a shift within type 2 subtypes that we see a complete disappearance of type 2X and become more to type 2A. So we've got some signaling that's driving some of these adaptations within type 2 fibers. So what's causing this? What's allowing us to see this adaptation to resistance exercise? And it's a balanced process. We've already mentioned in the first module of this class that, to see adaptation, we have to have a change in protein content. Proteins are driving the structural components and the functional components of all of our cells. And it's this balance between protein synthesis and muscle protein degradation. So current evidence suggests that in a non-disease state, the regulation of muscle mass is more importantly dependent on modifications of protein synthesis than protein degradation. So you think about the rehab process and you see skeletal muscle adaptation. That adaptation is because we're improving muscle protein synthesis, not because we've removed the degradation pathways. Now, there's part of that coming into play, and we'll briefly get into that here today, but not a lot, and more so on Thursday. But it's primarily we're talking about muscle protein synthesis. Now, we're not going to get into it here in this class, but we will in a few grad classes. If you start talking about atrophy, where you put someone on bed rest and say they're in the hospital and you see muscle loss there, there's actually quite a bit of heated debate on whether it's changes in synthesis or degradation. But when it comes to response to exercise and rehab, it's through the synthesis. And this rate is dependent on translational efficiency. So this is the protein synthesis per amount of messenger RNA. This is why we had to cover this stuff at the beginning of the semester. You've got this template of messenger RNA. Well, how much protein are you actually making from that messenger RNA? So that's translational efficiency. And then there's translational capacity. So you've got to have those ribosomes there, in a given amount for a given tissue, and they've got to be there in great enough quantity to convert that messenger RNA. So synthesis, translational efficiency, and translational capacity. So this is how efficient it is, and gives us kind of our peak capacity. So these are kind of some things that we've got to keep in mind as we work our way through now each of the things and each of the pathways that are going to drive some of this response that we see to resistance exercise. And we're going to start with really the basic one, which is just nutrition and muscle protein synthesis. So I've already mentioned this balance between synthesis and degradation. So I just want to make sure I've got that here. All right. So there's a fair bit of evidence that shows that circulation of anabolic nutrients, and leucine is the one that comes up the most, is sensed by this molecule complex called mTOR. It's the mammalian target of rapamycin. I'm going to always refer to it as mTOR. Everybody refers to it just as mTOR. If I ask you a question, I'll give it to you, as a full thing here. So we've got these anabolic nutrients like leucine that can be sensed by this mTOR, and that mTOR is going to come up quite a bit in lecture today. And that results in a considerable increase in muscle protein synthesis. both through enhanced translation initiation and elongation. So if you remember back to early in the semester, we talked about this, the initiation step and the elongation steps involved with translation. Those steps are what's targeted with leucine when you were talking about dietary supplementation of leucine and getting this increased muscle protein synthesis. So mTOR involved in the control of this translation initiation, and it does so by targeting some downstream molecules that aid in the binding of messenger RNA to the ribosomal pre-initiation complex. I'm not going to highlight that here because we're going to talk about that in a little bit more detail in a couple pages here. But that kind of gives you an idea of how things are going to progress. So let's just look at this graphically. So we've got our extracellular space, we have our intracellular space, here's our plasma membrane of our muscle fiber. So we've got, this extracellular essential amino acid, which is usually primary leucine. It's the one that's most studied. And this leucine can have a direct impact, it's not really highlighted very well, on this mTOR. And this mTOR then has an impact on these downstream targets, and we'll talk about 4-EBP1 and this initiating factor in a couple pages here, but that's all eliciting increases in muscle protein synthesis. And this is great. There are supplement companies that have made millions of dollars on this pathway, and good for them. But if you actually look at the literature, and the reason we're not going to talk about this pathway in much more detail is in reality, it's not a pathway that's that important, because if you actually look at the data, the response, and so the amino acid concentration that drives this muscle protein synthesis, cell and muscle, young and old subject, is found to be curvilinear. So there's a point at which more doesn't do you any good. And once you get to about the 10 grams of ingested essential amino acids, it doesn't seem to have that same stimulatory effect. So you start talking about someone who's on a very poor diet. This has come up with some of the early spaceflight literature that they realized that the diets they were giving these guys in flight wasn't sufficient. It was low in protein. And therefore, if they supplemented with a little bit of leucine, hey, things weren't quite as bad. And that's because they were in a deficit. You take and you kind of get above that 10 grams, it may not play a role. But if you're talking about an older adult who's maybe on a poor diet, this may be something that you can do in addition to the other pathways to drive some of this increase in muscle protein synthesis. So this is kind of our first step. Leucine, mTOR, muscle protein synthesis. Now, I've also got on here, there's other pathways, and you can see that they all kind of integrate with mTOR here. So let's talk about this insulin growth factor 1 pathway. And I generally refer to this as the IGF-AKT-mTOR axis, essentially highlighting this sequence of events. So insulin-like growth factor 1, or IGF-1, and again, I'll give you both the name and the abbreviation. Gets its name because it shares about 50% of the same amino acids as insulin. So therefore, it's like insulin. And it acts as a circulating hormone as well as a local growth factor. And as you might expect, because it's got a very similar amino acid identity to insulin itself, that it can interact with a variety of different types of receptors, and insulin itself can interact with a variety of different receptors. So here's insulin, here's IGF-1, there's other insulin-like growth factors, so here's IGF-2, and they all interact with, here's our insulin receptor, and here's our insulin-like growth factor receptor, so IGF-1R. So we see this in skeletal muscle, contains this receptors that bind to insulin. We've already mentioned that in the glucose lecture, and IGF-1 and or IGF-2. Now, you should hopefully start to recognize this receptor structure. It's got two alpha domains and two membrane-spanning beta domains. So we can hopefully, with some time here, you'll start recognizing the kind of pathway that's going to follow this type of receptor. So it's a tyrosine kinase self-phosphorylating receptor. So skeletal muscle's got this type 1 insulin-like growth factor receptor, so IGF-1R. Why do we care about this pathway? Well, we know this pathway specifically regulates aspects of things like satellite cell-driven myogenesis, such as proliferation, differentiation, and fusion. So it's a key step in not only satellite cell-driven processes, but as we'll see here on the next page, actual adaptations with the skeletal muscle response. So I've got this figure here, and I'm going to use the figures on the next slide, but this kind of just highlights this, these pathways and what they all drive. Nothing from this particular figure, though, is going to be on the exam. Okay, so what does this IGF-1 receptor do? And it activates a variety of things. Again, this is why signaling pathways, I think, can get really fascinating, is that you stimulate one receptor and it can have a cascade of events and really amplify the signal. So I'll let you write these down before I talk about them a little bit. So we've got three primary pathways when we're dealing with muscle plasticity and strengthening through this IGF-1 receptor. The first is the PI3K or phosphoinositide 3 kinase. I'm just going to say PI3K. So it's the PI3K AKT mTOR pathway. That's the primary one. Main pathway through which muscle protein synthesis is stimulated in response to IGF-1 and does appear to respond in some ways to mechanical activity, so the deformation of the cell membrane that come with muscle contraction. We're also going to have the ERK MAP kinase pathways. So remember we talked about the MAP kinase pathways associated with these tyrosine kinase receptors. And then there's also a protein kinase C pathway. We'll talk about the first two in a little bit of detail, and not so much the PKC, but know that these three steps or three types of pathways are functions of IGF-1 receptors. We'll talk about them in a little bit more detail here, but I want to at least go over them just briefly here with the figure, and then I'll give you the text, and then I'll give you another figure to kind of reinforce what's happening. So here's our extracellular space, here's our cytoplasm, here's our plasma membrane, down here's our nucleus. Here's IGF-1. Here's our IGF-1 receptor. So we get binding to the receptor. And hopefully you've just with a little bit of initial studying from last semester or last module, you're starting to recognize some of these terms and what the steps are with those terms. So if you look at this, you can say, okay, well, here's the IGF-1 receptor. I know it's a tyrosine kinase just based off its shape. Well, there's insulin IRS1 donking, there's PI3K, there's AKT, and here's mTOR, and protein synthesis response, all through this step here. And I've highlighted kind of the key steps, but we've talked about going from PIP2 to PIP3. We'll talk about this step here in just a minute, but we get this basic signaling pathway that we learned prior to spring break, driving protein synthesis in response to exercise. The MAP kinase pathway. So here's our growth-like receptor 1. We get RAS, and then we get our MAP kinase pathway. So we talked about the ER1/2 as one of our MAP kinases. That actually then, as we kind of talked about this pathway doing, has a direct impact on the nucleus and increases gene expression and increases cell proliferation. So this one receptor is driving the adaptation to exercise in two different ways. Direct effect on the protein synthesis through mTOR, and then it also can impact gene expression. So what do you need to really know? Oh yes, question.
What is an example of like of an increased gene expression? What gene would be being expressed?
So that would be, think about the response. So if we're talking skeletal muscle here, you know, if you're talking about an increase in cross-sectional area, what do you need? I take a muscle fiber from this person who's untrained and this person who's untrained. What are some of the proteins that I would expect to see more of in here? More actin, more myosin, more of the enzymes that are going to be associated with that. All of those have some gene that had to get turned on in order to make the messenger RNA that would then make the protein. So all of those genes expressions would be ones that could be potentially targeted. You know, part of it is we're not going to get into exactly which genes because we're kind of still trying to figure some of that particular stuff out. But if you want to talk about an increase in cross-sectional area, so you're talking about sarcomeres put in parallel, yeah, you're going to need more actin, more myosin, more troponin. All of those things are going to have to be more proteins. And if you can increase the gene expression for those proteins, that kind of is where that dogma, we had to talk about that dogma, the central dogma of DNA. So basically we're transcribing DNA to messenger RNA through this gene expression. And once we get that messenger RNA can get out here, and then through this pathway, convert that messenger RNA to protein. So it's really a double barrel response here. All kind of initiating in part through this IGF-1 receptor. Okay, so what are the real steps that you need to know? There's a lot of steps here, but I want to break it down to the key sequence of events that take place that I would potentially test you over. So step one, we get binding between our receptor and our growth hormone. So here's insulin growth factor 1 receptor binds to insulin-like growth factor 1. So we get that binding. So that causes the receptor, and I'm going to highlight kind of the key steps, but not everything in some of these sentences because they're kind of explanatory. So we get binding, we get conformational change. That leads to activation of the tyrosine kinase. Remember, we talked about the different types of receptors, and tyrosine kinase receptors was one of those, and it had this basic structure. Okay, so because it's a tyrosine kinase, we know that phosphorylation gives us a docking site for signaling proteins, such as the insulin receptor substrate, the IRS1, that we learned about in Module 2. So I'm going to scroll down and just look at this up to this point in our figure here. And then I'll scroll back up. So here's our receptor. I mentioned there's alpha and beta components to it. Here's our insulin-like growth factor 1. So this is our IR1 receptor. It gets phosphorylation. Here's our IRS docking protein. And all this allows it to get to this point here. So basically, up to this point is point 13. This docking protein, so this insulin receptor substrate 1, elicits PI3 mediated increases in this PIP3. And I'm only highlighting up to this PIP3 because that's as far as you need to know. You don't need to know its actual name. the PIP3, oh, go away, leads to recruitment of AKT, which again, if you look at some of the older literature, you'll see it as protein kinase B. I'm just highlighting AKT. And it gets phosphorylated, subsequent phosphorylation. So look at our figure again. So we've got step 1 here is binding. We get some phosphorylation. We get our IRS. That activates the PI3 kinase. And it lets us get this conversion, so it drives it from left to right here. Here's our PIP3. What activates and phosphorylates AKT. So, how am I going to ask you a question? It's very similar to what I did on the last exam. Put them in order. I'm going to give them to you. You need to know the order in which everything occurs. Okay, so what does AKT do? And this is where it gets, hopefully you're thinking a little bit about, well, I've already learned that if insulin itself binds to an insulin receptor, it upregulates GLUT4 translocation through AKT. So is this the same AKT? Is it going to do that same thing? And there's variations in the AKT molecule within skeletal muscle. It's a family. We like to say, as physiologists, that, we say AKT, but we don't get much very specific from there. It's like, say, call someone a basketball player, but there's different positions, same with football and everything else. So there's these different, subcomponents of this AKT family. In human skeletal muscle, AKT1 is involved in muscle hypertrophy, while AKT2 plays a role in insulin-stimulated glucose uptake. So while we just say AKT, and this would make great true or false type question, but just to understand that there's subtypes. We're not getting into those subtypes here. So since we're talking about skeletal muscle hypertrophy, this insulin-like growth factor 1 drives increases in AKT1, which is going to give us subsequent muscle hypertrophy. So what does it do? How does it have its actual action? An important mode of this AKT1 is it inhibits this TSC2, which is known as tuberous sclerosis protein 2, as part of this complex. I'm going to highlight point 17, and then I'm going to go to the figure because I think that illustrates it a little bit better than the text here. This inhibition of TSC2 allows Reb to be in its GTP form, which allows it to activate mTOR1 complexes. All right, I'll come back up to this a little bit. Well, let's look at this, so here's our AKT. It's been phosphorylated, it's upregulated. It inhibits the TSC complex, so that's where this little marker here is. This inhibits this, which prevents this complex, or removes the inhibition of this complex on Reb GTP. So the way I like to think about this is kids on the playground. In this complex is the bully on the playground, and it's keeping Reb over here in this corner where it can't do anything. It's inactive here. This is the bully. It's preventing Reb from doing what it wants to do. AKT is the school teacher. When it gets activated, comes in here, inhibits the bully from being a bully, and allows Reb to be in its active state, which then activates mTOR, which initiates some this sequence of events here we'll talk about next, the initiation and elongation steps. So that's what I'm talking about here, is that AKT, activated AKT, inhibits the bully that is the TSC complex. If we inhibit TSC, so we inhibit the bully, it allows Reb to be in its active form, which allows it to activate mTOR. There's also this parallel complex or site signaling where AKT inhibits the induction of atrophy signaling through inactivation of this FOXO, which is forkhead FOXO. So it's not only stimulating protein synthesis, but it's preventing the degradation. So it's preventing atrophy. So it's kind of working on both sides of that balance. All right, so we've got AKT through this pathway, gets a mTOR. And let's go from there. Another pathway through AKT and this PI3 kinase axis is through this GSK3 beta, which has an inhibitory effect on initiating factor 2B. I'm not going to highlight this. This is me getting a little excited when I'm writing notes. This is cool, and it's great for you to know, so that's why I kind of left it on here, but I'm not going to highlight it. I'm more interested in you knowing these other pathways, so not highlighting it because I'm not going to ask you about it, but I'll talk about it just briefly. This pathway, this IGF-1 pathway is pretty important because we can see through AKT, this thing's having a pretty big impact. It's impacting mTOR. Here's our TSC complex. It's driving protein synthesis and protein degradation. So here's the FOXO. It blocks FOXO and prevents protein from being degraded. It activates mTOR, these initiating factors. So remember we talked about the EIF portion of these molecules. We know just because of the EIF that they're initiating factors. So it's driving initiation of translation and it's preventing degradation. And this point 19 just talks about this sequence here. But again, I'm highlighting it here because I'm talking about it, but you don't need to know this sequence for the exam. What you do need to know is the MAP kinase signaling, because it's also pretty important in driving these adaptations to muscular contraction. So remember I mentioned that MAP kinase, there's subtypes of this MAP kinase, kinase, MAP kinase, MAP kinase sequence of events. And the most popular is this ERK1/2 MAP kinase signaling. It increases in response to muscular contraction. And it works in a very similar manner as AKT, in that it walks into the playground and prevents the bully from having its effect. So it inactivates and prevents this TSC2 from inactivating Reb. So remember, this TSC2 hits the bully. It's preventing the activation of mTOR. Both AKT and ERK1/2 come in and they basically prevent this bully from knocking down mTOR. Allows mTOR to become active and have its effect. So you can see here this mTOR is a pretty key regulator. And if you really get into much of the literature on skeletal muscle adaptation, whether it's changes in response to exercise and rehab or changes in some cases to dysfunction or disuse, you're going to come across mTOR. It's going to be mentioned. So what is actually, we've talked about everything up to mTOR. What are these sequence of events happening down here? And how do they relate with what we've talked about so far in the class? So mTOR. It's a key regulator of cell proliferation, cell growth, differentiation, protein synthesis, cytoskeletal organization, energy homeostasis, and substrate metabolism. All things you would expect to be modified in response to resistance exercise. So this is why it gets brought up a lot. It's a key regulator. We had to talk about the steps that drove this increase in mTOR activity. Now we'll talk just briefly about what is mTOR actually doing within the skeletal muscle to give us these responses. You'll see a little bit of two types of mTOR, mTOR1 and mTOR2. mTOR1 is really the key one when we're talking about skeletal adaptation to resistance exercise and nutrition. It's the kind of critical control. mTOR2, you'll see a little bit more with glucose uptake, but we're just going to focus on mTOR1. So, I like figures and I like pounding them into your head. So here's our insulin growth factor. Here's our growth factor-like receptor. We get our IRS docking. We get, can't see that. Here's PI3K, gives us PIP2, gives us AKT. AKT inhibits this TSC1, TSC2 complex, so removes the bully. So that allows Reb to activate mTOR. Leucine, so if we are deficient and needing this increased leucine or amino acid supplementation, it's going to come in through here and have a direct impact on mTOR. And we're going to see then changes in initiation and elongation. And there's a lot of different steps between mTOR and in this process here. I'm going to talk about this pathway here in particular, because it relates very well to what we've talked about in the first module of class. So I highlighted it above. mTOR promotes translational initiation and elongation, resulting in protein synthesis. It promotes translation via the phosphorylation of 4E-BP1. And I'll show you a figure of this, don't freak out. Phosphorylates 4E-BP1, which detaches from this initiating factor, allows the ribosome to bind to messenger RNA and initiate translation. And I'll show you a figure, and this is actually one we used in a previous class. I'll start with just the image here, and I'll come back up and highlight. So we've got really two steps here. So here's our initiating factor. I apologize, it's a little fuzzy here. So here's our IF initiating factor. Here's this 4E-BP. Right next to it, inactive. So this is in an inactive state here. It can't do anything. It is worthless. If we activate mTOR, so through leucine, through insulin-like 1 growth factor, that allows these two to separate. So here's our phosphorylation. So it breaks them up. So mTOR comes in, breaks them up, allows this initiating factor to come and bind. So we talked about our small and large ribosomal subunits. It allows for initiating. So we get our initiating factor here, and it can come in and do what it needs to do, and we can get initiation and elongation of our amino acid chain. And that's really what's just stated here in this first step. So our initiating factor that's in its inactive form because it's bound to this 4E-BP, it's released once mTOR comes in and phosphorylates it, and allows it to bind to messenger RNA and results in recruitment of our ribosomal subunit. Again, it's going to be about putting them in sequence. You're not going to have to pull these steps out of thin air. Know the sequence at which these steps occur. That's the key thing. So I'm going to pull this stuff and just put them in a list, and you need to put them in order. And there's a good chance that I'm going to throw a couple things in there that wouldn't be part of that pathway. And you'll have to identify it as not part of the pathway. So if you know the sequence of events, you're going to be good. You don't have to pull this stuff out of thin air. You take an advanced molecular phys class, then you will. But it's about the pathways. So there's our basic sequence of events. So step 1, they're bound. Step 2, mTOR. Step 3, they separate. Step 4, they become initiated. Subodine did some work, as well as some others, to kind of validate it, that there's also a strong association with just the mechanical loading an mTOR. Now, that's about all we know at this point, that there's these receptors and changes in things like the contractions and calcium signaling, but just the mechanical loading itself, through some pathway that we're not really sure about, activates mTOR. So there's some sensing of the mechanical load placed on skeletal muscle. So that mechanical loading is sufficient to activate mTOR in a growth factor independent manner. And they can test this in a variety of ways. We can apply a drug that's going to block to this receptor. So we can give we talked about agonists and antagonists. I can give an antagonist that's going to bind to this IGF receptor and not do anything. It's just going to block it. And then I can stretch the muscle and I can activate mTOR. And that's through an independent pathway. If I prevent this step from happening, I know that the mechanical load itself. We're not going to talk about it in this class, but hopefully you're starting to piece together, there's a lot of different ways that which I can stimulate skeletal muscle adaptation. If I can find exercises that increase insulin growth factor 1, I'm going to see adaptation. If I do things that increase the mechanical load placed on the muscle, that's also going to play a role. And you can start, tailoring a type of exercise or rehab to get us this protein synthesis. Okay. Another thing that comes up when you start talking about skeletal muscle adaptation is myostatin. It's also been called growth and differentiation factor 8. And it is a regulator of muscle mass. So I'm going to just highlight. Myostatin functions as a regulator of muscle mass. And it got some really good excitement built around it from some animal studies. So they use transgenic mice. Basically, they breed these mice to lack myostatin. And if we look at these mice, so here's our wild type mouse, and then myostatin deficient is the FF. So this is cross-sectional area unit per muscle fiber, cross-sectional area per micronucleus. We can see pretty significant increases in muscle fiber size in these deficient mice. So they lack myostatin, nothing else. They're not trained, just lacking myostatin. We see this profound increase, greater than in some cases what you'd see with resistance exercise training in these animals. If we look at it kind of as a percentage, so this is percent of normal muscle to body mass ratio in these mice, as a percentage of the mean normal myostatin messenger RNA expression. So here's our normal wild type mice where they've got somewhere a range of myostatin. And then if we start removing that myostatin, we start seeing kind of a threshold here where we start seeing dramatic increases in muscle per body mass. So really highlighted that, okay, this myostatin might be playing a pretty key role in these skeletal muscle adaptations. So what is it actually doing? So it appears as a secreted factor that negatively regulates muscle size and is a key molecule in muscle wasting. And it does so by negatively regulating the AKT-mTOR pathway that we just learned about. So here's our myostatin. So I'll highlight this up here. So we have, it's a secreted factor. So here's our myostatin. We're not going to really worry about its receptor at this point, but we can see that it blocks, it prevents, it stops this pathway. So it's going to negatively regulate AKT. So if we've removed activation of AKT, we're now not going to get this pathway, and therefore we're not going to get the initiation and elongation. Yeah.
So like when people take like a statin or other statins and like muscle contractions, is that because they're structurally similar?
Yeah, I think that is, I don't know for certain, so I don't quote me on this, but I believe that there very well could be some crosstalk between some of those pharmacological agents and myostatin. That would be interesting. Has anybody seen any of that? That are just on kind of a lipid on a regular statin. Yeah, and that very well could be through this pathway. I don't know. I'm gonna have to look. That's now intriguing, because we talk about Brad and I just put in a big grant looking at statins. There's an idea that statins could be protective of the cardiovascular system in cancer patients. But if there's also cancer cachexia, that might have a worse effect on skeletal muscle. So that's really cool. I'll have to look into that. But it would be interesting to know if this myostatin is structurally similar to like the statins you would give for cholesterol. If so, it would be negatively regulating this pathway here, which would prevent this protein synthesis and promote wasting. I do know in response to exercise, so this is from muscle biopsy. So we've got resistance exercise and running exercise, and we talk about fold change. So this is a single bout of exercise, suggesting it may play some role. There's really conflicting data in response to just training, how big a role myostatin plays. If we just look in response to individual bouts of exercise, so times 0, 1 hour post, 4 hours post, and we start seeing a negative regulation of mTOR over the first 24 hours in response to resistance versus running. We can see a pretty significant impact in resistance exercise at about the 8-hour point. So suggesting it may have some role with resistance exercise, but it doesn't appear to be the primary role. The other thing that comes up when you start talking about skeletal muscle adaptation is satellite cells and the role in muscle growth and muscle repair. Again, this is kind of one of those more ongoing areas of research, so there's not a lot of depth here, because there is some controversy about what's going on. But we do know there's repair and growth is dependent on these myogenic precursor cells called satellite cells. And they're really the stem cell of muscle tissue. When we find them between the basal lamina and the sarcolemma, the myofiber, so we've got damaged myofiber, it can activate, so we can have a quiescent satellite cell, we can get it activated, and we can start seeing some regeneration. The key things that we do know is that we can activate these satellite cells through insulin-like growth factor. So it's likely going to have an AKT-mediated pathway, nitric oxide, and BMP, so bone morphogenic protein. All have been reported in the literature to be involved in the activation. What's actually happening with these cells is there appears to be two signaling pathways, the Notch and the WNT signaling pathways, when it comes to growth and repair to actual muscle damage. I'm just going to highlight these two. They make nice true and false type questions. So Notch is a receptor. Ligands trigger the cleavage of its intracellular domain. So here's our Notch signaling over here. So here's our Notch. We get some ligand that binds. It's got an intracellular domain that pops off, translocates to the nucleus, and gives us our differentiation of our satellite cells. The WNT signaling, we see this following muscle injury response is associated with differentiation of satellite cells. So here's our WNT, got some known receptor here, and differentiation. The literature with this is really hard to interpret, because I've reviewed a few papers that have looked at muscle damage and some of these signaling pathways. And the damage that they do is insane. I mean, it's crazy. And they do it in animals, and it's damage that you would not expect in any human population. I mean, unless it's the most extreme. So in order for them to see some of these responses, they'll do like massive freeze protocols, they'll do massive eccentric exercise and see just profound muscle injury to see some of these responses. So it's really hard to truly understand what's going on here because, while there is some muscle damage associated with resistance exercise, it's profoundly less than what you see in some of these experimental models. So as such, poorly understood how these mechanisms operate here in physiological muscle hypertrophy. They may play a role, they may not. It's really hard to tell what's going on. But we do know if you've got muscle damage, even if it's extreme, these two pathways come into play. There's our differentiation of satellite cells. All right, so in summary, unlike endurance training, which mainly gives us a response through gene expression level, so transcriptional changes, strength type training is more associated with translation. Key pathways and major integrations of the signaling cascade is through mTOR, growth factors acting through the IGF receptor, AKT pathway, and through mechanical stress. But again, there's that major integrator of mTOR. And when mTOR is activated, it increases protein synthesis through both translational initiation and elongation.